am Serena, Assistant Professor, Department of Microbiology from E-Content Development Center, Majlis Arts and Science College, Puramannur. Today in this section, I would like to share some identical features of a bacterial species known as Salmonella Typhi. As we know that our environment consists of large number of microorganisms like protozoa, bacteria, fungus, virus, etc. Among them, bacteria is the most predominant one, which causes human infection as well as animal infections. Here we have an introduction to Salmonella genus. This Salmonella genus coming under a family known as Enterobacteriaceae. And Salmonella are the human parasite, or they are the parasite of human as well as animal intestine. And the most important member of this Salmonella genus is Salmonella Typhi, which is a human pathogen that causes a enteric fever known as typhoid fever in humans. Then this Salmonella Typhi is first described by a scientist known as Eberth in 1880, and it is first isolated in pure culture by Gafke in 1884. Hence, the Salmonella Typhi is known as Eberth Gafke bacilli or Eberthella typhi. Then this Salmonella are classified into two major groups, that is typhoidal group as well as non-typhoidal group. Typhoidal group is also known as enteric fever groups. The organism coming under this group causes infection in humans, that is they are the normal parasite of humans, and they cause an infection known as typhoid fever, and also this organism coming under this group causes another infection known as paratyphoid fever. Then typhoid fever is caused by Salmonella typhi and paratyphoid fever is caused by Salmonella paratyphi. Again, Salmonella paratyphi are of three types. They are Salmonella paratyphi A, Salmonella paratyphi B and Salmonella paratyphi C. Then another group is non-typhoidal group. They are also known as food poisoning group. They are the normal parasites of animals, but they can cause infection in humans also and producing gastroenteritis, septicemia and some localized separative lesions. Then next is some identical features of Salmonella typhi. Here Salmonella typhi is a gram-negative rods and have 1 to 3 into 0.5 micrometer length and breadth respectively. Then they are motile with peritrichous flagella. They are the locomotory organelles present all over the body of Salmonella cells. Then they are capsulated, that is capsule is present outside the cell wall, and you can see here this nucleic acid, ribosome, then plasmid are present in the Salmonella typhi. Then move on cultural characteristics. Next is about their cultural characteristics. Cultural characteristics means what are the important factors or conditions should be provided for their maximum or optimum growth. Here Salmonella typhi is aerobic as well as facultative anaerobes. Aerobic means they can grow in the presence of oxygen, but they can also grow in the absence of oxygen, hence they are known as facultative anaerobes. Then they can grow in the temperature range between 15 to 41 degrees Celsius and their optimum may be 37 degrees Celsius, that is 37 is the optimum temperature at which their maximum growth occur. Then their pH may be 6 to 8, that is the optimum pH required for their maximum or optimum growth. Then this Salmonella typhi can grow on ordinary medias like nutrient agar and produces large, loose, low convex, translucent colony, and on McConkey agar they produces pale colored colony or colorless colony, that is due to non-lactose fermentation. Then on some selective medias like deoxycholide citrate agar and XLD, XLD means xylose lysine deoxycholide agar, they produces black colored colony with the production of H2S gas. Then on Wilson's and Blair bismuth media, they produces black heads with a metallic sheen due to the production of H2S gas. Then this Salmonella typhi can be enriched by growing them in enrichment media like selenate F as well as tetrathionate broth. These are about cultural characteristics of Salmonella typhi. Then next is about their biochemical reactions. Biochemical characters are used to identify the bacterial species. Here Salmonella typhi have ability to ferment number of sugars like glucose, maltose and mannitol with the production of acid and gas, but they cannot ferment lactose, sucrose and salicin. Then about their in-vivo reaction, the Salmonella typhi not produce indole, that is indole test is negative, then methyl red test is positive, Voges-Proskar test is negative and citrate utilization test is negative. Then Salmonella typhi have ability to produce H2S gas except Salmonella paratyphi A. Then Salmonella typhi does not have ability to hydrolyze urea because they do not produce urease enzyme that hydrolyze the urea. Then move on resistance. Here Salmonella typhi are delicate organism. They can be easily destroyed by heating or boiling at 55 degrees Celsius for 1 hour or at 60 degrees Celsius for 15 minutes. Then they can be easily destroyed by pasteurization as well as heating or boiling. Then Salmonella typhi remain viable for weeks in polluted water and soil. Then remain viable for several weeks or months in ice crystals. Then when we prevent them from drying, they can remain viable or active for several years. Then this Salmonella typhi are sensitive to mercury chloride as well as 5% phenol. Then next we have look at the antigenic structures of Salmonella typhi. This Salmonella typhi possesses 3 types of antigens. They are flagellar H antigen, somatic O antigen, then surface VI antigen. This H antigen present in the flagella, then O antigen present in the cell wall, that is the integral part of the cell wall, and VI antigen present in the capsule, that is capsular antigen, which is confined to virulence. Then first of all H antigen. This H antigen is present on flagella of Salmonella typhi. This is protein in nature and heat labile. So it can be destroyed by boiling or treatment with the alcohol, but not by the treatment with the formaldehyde. Then when mixed with the anti-serum, that is anti-serum means the serum containing antibody against H antigen, mixed with H suspension can agglutinate rapidly and produce large, loose, fluffy agglutinate or clumps at 37 degrees Celsius. Then this H antigen is strongly immunogenic, that means have ability to induce the production of antibody, and this antibody production is in high titer following infection and immunization. And this H antigen is used to detect Salmonella typhi basically in this Vidal test or other diagnostic reactions. Then next type of antigen is O antigen, which is present in the cell wall, that means they are the integral part of the cell wall, which is phospholipid protein polysaccharide complex. And this O antigen is identical to endotoxin, which can be isolated or extracted from the bacterial cell by the treatment with the trichloroacetic acid. And this extraction method is first described by the scientist Boivin, hence this O antigen is also known as Boivin antigen. And this O antigen is resistant to boiling, then other acids as well as alcohol. Then this O antigen is less immunogenic, that means this O antigen induces the production of low amount or low titer of antibody. Then when mixed with the O antiserum, that means antiserum containing antibody against O antigen, this O antigen or O suspension agglutinates and form compact jockey as well as granular clumps at a 50 to 55 degree Celsius. Then this O antigen also used to identify Salmonella typhi in diagnostic method like Vidal test. Then next type of antigen is VI antigen. This VI antigen is present in the capsule since it is confined to virulence of the Salmonella typhi. And this VI antigen act as a virulent factor by inhibiting phagocytosis, resisting complement fixation or complement activation as well as bacterial lysis. Then VI antigen is polysaccharide in nature and heat labile, can be destroyed by treatment with HCl or NaOH, but they unaffected by alcohol and formaldehyde. Then this VI antigen present in the capsule, hence they enclose or mask the O antigen. So, if the culture containing VI antigen that not agglutinable with the O antiserum since it mask or prevent the O antigen. This culture become only agglutinable by boiling or heating at 60 degrees Celsius for 1 hour. Then VI antigen is poorly immunogenic, that means that induces antibody production in low titer. Then this VI antigen is not used in Vidal test because this antibody against VI antigen present only in the early phase of the infection. Then they disappear thereafter, hence they are not used in the Vidal test or diagnosis of typhoid fever. That is all about some features of Salmonella typhi. We will continue with the remaining features of Salmonella typhi in the next session. Thank you.